Okay, I apologise for the abrupt ending to the previous video. I didn't foresee the fire alarm going off. Right, okay, so we were discussing the Wnt beta-catenin pathway. So you have the TCF-LEF family transcription factors binding to the Wnt response element here, and then you have this Groucho TLE family protein binding on top of that T cell factor, lymphoid enhancer binding factor, family transcription factor. And we were just discussing that TLE stands for transducin-like enhancer, and then some people will add on the off-split protein. Some people just call it a transducin-like enhancer. Okay, right. One of these family of proteins is going to bind on top of the T cell factor / lymphoid enhancer binding factor, transcription factor, when there is no beta-catenin. And I want to stress this: when beta-catenin is present, this protein is not going to get a look in, basically. It's not going to be allowed to bind here. It can only bind here when there's no beta-catenin. Okay, so when there's no beta-catenin and we're in the off state, we get this Groucho TLE family protein binding on top of the TCF-LEF family transcription factor. And then, what's going to bind on top of this? Well, an enzyme is going to bind on top of this Groucho TLE family protein. Okay, and this enzyme is what's going to be known as a histone deacetylase, which is often abbreviated to an HDAC, like so. So the H stands for histone, and then the DAC stands for D, and then acetylase. Okay, so the D is for D, and then the AC is for acetylase. Okay, so let's just highlight this up. So the H is for histone, the D here is for the D, okay, and the acetylase then, which I won't do in pink, I'll do in red, the acetylase is for AC. Okay, so histone deacetylase here. Okay, and we'll highlight this up in R. So there are many histone deacetylase enzymes, but we're just keeping this general. A histone deacetylase is going to bind on top of the Groucho TLE family protein. Now, this is going to remove acetyl groups from histone proteins. Okay, so in order to understand the significance of that, I'm going to now discuss histone proteins with you to make sure that everyone's on an equal footing with regards to their understanding of histones. Okay, so let's discuss histones then. So, there are 5 families of histone proteins within humans. Okay, there is the histone 1 family of histone proteins. There is the histone 2A family of histone proteins. There is the histone 2B family of histone proteins. There is the histone 3 family of histone proteins, and then finally there is the histone 4 family of histone proteins. So I want to stress that these are not just individual proteins. H1 is not an individual protein. Again, it's a whole family of proteins. It's the same for all of these. They are families of proteins. So histone 2A, for instance, will contain absolutely loads of different proteins. However, all of these proteins perform the same function, and therefore they're all grouped into this family. Okay, right, so what is the function of histones? Well, basically, histone proteins are going to bind together in these structures known as histone core complexes, which are basically octamers of histone proteins. So let me draw this here. Okay, so I'm going to draw it as a cube. Right, so this is the histone core complex here. Okay, and basically it is an octamer of 8 separate histone proteins bound together. So let's split it up to make it obvious that it is made up of 8 separate portions here. Okay, so these lines are going to divide up the histone core complex into these 8 separate parts. So it is an octamer then. Okay, so we can see 7 of these portions. 1, 2, 3, 4, 5, 6, 7. The final, 8th is right at the back there, okay, so we can't see it. Okay, so this is our histone core complex made up of 8 histones overall. Now, this histone core complex is going to be made up of overall 2 histone 2A family histones, 2 histone 2B family histones, 2 histone 3 family histones, and 2 histone 4 family histones. Okay, so let's now show where these are going to be. So this one right at the start here, this is going to be a histone 4 family histone. Okay, so I'll highlight this up in blue. So when I put an H4 in there, that doesn't mean a specific one of the histone 4 proteins. That just means it is one of these family proteins, because remember, they all do the same thing as far as the histone core complex is concerned. So it doesn't matter which one you put there, basically. Okay, then we're going to have our other histone 4 protein down here. Okay, like so. Now, I want to next show the histone 3 proteins on this diagram. So we'll have a histone 3 family histone there, and we'll have another histone 3 family histone up there. Okay, so I'll colour in these histone 3 proteins in red here. So this is one histone 3 family protein, and here is another histone 3 family protein there. Okay, so those are H3s. Now, let's discuss this further, because when you actually assemble a histone core complex, when you assemble one of these octamers, you do not just smash 8 histone proteins together and hope that it's going to just form an octamer. Instead, what you actually do is you bring parts that you've made already, basically. And one of the parts that you have made already is the portion that we've now coloured in. Okay, so it is this tetramer, meaning a 4-membered structure, containing 2 histone 3 histones and 2 histone 4 family histones, like so. Okay, so we'll once again colour the histone 4 proteins in blue here, and then we'll have the histone 3 proteins in red here. Okay, so this structure, this is known as a histone 3, histone 4 tetramer. Okay, so when constructing a histone core complex octamer, you start off with that. Okay, so imagine the portion that we haven't coloured in yet is not there. This portion here is the histone 3, histone 4 tetramer. You're going to bring this in as one of the things that you've made earlier, basically. Okay, right, now let's discuss the other side, the other half. Okay, so this is going to consist of 2 histone 2A proteins, so we're going to have one of those here at the front. Okay, so we'll colour this histone 2A protein here in orange. Okay, and then we'll have, well, we can't actually see the other histone 2A. The other histone 2A will be right at the back. It's the one that we can't see. Okay, the other remaining 2 that we can see, this one here and this one here, those are both going to be histone 2Bs. Okay, so this is a histone 2A here in orange, and these final 2 here, one there and one right at the front here, those are histone 2Bs. Okay, right, so once again going back to the construction of a histone core complex octamer like this, basically the histone 2As and the histone 2Bs come pre-assembled into dimers. So they're not quite pre-assembled into tetramers, but they're pre-assembled into dimers. So you have a histone 2A in orange here, bound to a histone 2B, which will be in vivid purple here. Okay, and this structure is known as a histone 2A, histone 2B dimer. Okay, right. So, when constructing then a histone core complex, just to review the whole story now, what you do is you take a histone 3, histone 4 tetramer, which makes up half of the octamer already, and then you bring in 2 histone 2A, histone 2B dimers and stick them onto that, and that produces you a histone core complex. Okay, so that's just the histone core complex. That doesn't tell us anything about what this thing actually really does. Okay, so what does it actually do then? Well, basically, DNA in the nucleus of human cells is not just free. We are not bacteria, okay? Our DNA is wrapped around proteins, basically. Okay, so what's going to happen is these histone core complexes are the structures that we wrap DNA around. So this green line is going to represent the double strand of DNA, so it's representing a DNA duplex. It will wrap around the histone core complex like so. Okay, so it's wrapped around once now, and that's going to go round again. Okay, so round it goes again, and then it comes off again. So it wraps around the histone core complex twice, like so. So that's the histone core complex, and we've seen the role of all of these 4 families of histone proteins. And I'll stress once again that I have written the family members that you will have, okay? I have not specified any specific proteins here. You can have any old histone 2A family member here, any histone 4 family member here, and the same for all the other 6 positions, okay? So this just tells you which family the histone at each position is, okay? So all the histones that are in each of these families all perform the same function, basically. Okay, right. Let's now move to the final family, which we haven't actually mentioned anything about, which is the histone 1 family. Okay, so what does the histone 1 family proteins do? Well, they bind to the incoming DNA and also the outcoming DNA. So here's the DNA coming into the histone core complex. Here's it going out of the histone core complex. Basically, histone 1 proteins bind to these 2 strands and hold them together, and that secures the whole arrangement that we have got here. Okay, so this is a histone 1 protein here. Okay, right, so that's how DNA wraps around histones. I now want to discuss a few more pieces of terminology just to make the picture complete, which are chromatin and also the concept of a nucleosome. Okay, I'm just wondering if I've got room to show this here. I think I'll go on to another picture. Okay, right. Chromatin then. What is chromatin? So basically, DNA is wrapped around histones continuously. Okay, so here let's say this is a histone core complex, and here comes the DNA strand. It will wrap around this histone core complex. There's once, here's twice. Then it will move along down to the next histone core complex, which will be here. Okay, it will then wrap around the histone core complex once, it will then wrap around the histone core complex a second time, and then it will go along to the next histone core complex. So we continue this process on and on and on. So this rectangle here, this now represents a histone core complex. Okay, and the DNA wraps twice around it, and then you've got this linker piece of DNA that is between 2 histone core complexes here. So this is linker DNA. Okay, this is what chromatin is. This strand where you've got DNA wrapped around histone core complexes. It's often compared to beads on a string. The string is like the linker DNA, and the beads are like the histone core complexes with DNA wrapped around them. Okay, so that strand is chromatin. Okay, right, so our nucleus is therefore full of chromatin, which is this strand of DNA wrapped around histone core complexes continuously. Okay, right, the final concept is the concept of a nucleosome. Now, a nucleosome is the repeating unit of chromatin, rather. Okay, so basically, if we look at chromatin, what is the repeating unit of chromatin? Okay, well, people often mistakenly say that a nucleosome is the histone core complex with DNA wrapped around, but that's not true because, well, not quite, it's close, but it's not quite true, because if you just have that, you don't have chromatin. Chromatin has to include the linker DNA as well. So in fact, a nucleosome consists of the histone core complex with the DNA wrapped around that histone core complex, which I'll show in orange here, okay, along with the linker DNA that takes one to the next histone core complex. That is the repeating structure of chromatin. You can repeat this multiple times, and indeed that is exactly what you repeat multiple times to produce a piece of chromatin. Okay, right, so that's the concept of chromatin and the concept of a nucleosome. Right, so that was a little bit of a deviance into a bit of genetics here, or rather epigenetics. Okay, let's now, well, actually, we haven't talked about the epigenetics yet, so let's now talk about what acetylation of histones is going to do, and more specifically, what deacetylation of histones is going to do, because that's what we've got now. We've got an enzyme, a histone deacetylase, which is going to deacetylate histones. So, basically, most of your DNA is actually going to be wrapped around histone core complexes. This is the way DNA exists in your in the nuclei of your cells. It is just a string like this that goes on and on and on. So most of the length of the DNA is actually wrapped around histone core complexes, and very little of it is in the linker DNA between histone core complexes. So that means that your genes are going to be wrapped around histone core complexes. Now, you might be thinking, isn't that going to make it awfully difficult for enzymes such as RNA polymerase II, which is a massive great complex of proteins, to gain access to the DNA? Because, you know, if it's tightly wrapped around that histone core complex, it's going to make it awfully difficult. Okay, and indeed that's exactly correct. It does make it more difficult. So, if you actually want to use a gene, you have to have that portion of the DNA wrapped loosely around the histone core complex. Okay? And the way that you can make the DNA wrap loosely around the histone core complex is by adding groups onto residues within the histone proteins. Okay, specifically, there are two groups that are going to be important for our discussion, which you can add onto lysine residues within histone proteins. Okay, so I'm just going to discuss the structure of lysine, and then we'll discuss these groups that can be added onto lysine residues within histone proteins. Okay, so we'll start with the core amino acid residue structure. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group, and then the R group of lysine consists of 4 methylene groups. And because I don't want to have to draw 4 methylene groups, what you can do is you can bracket this single methylene group that I've drawn here, subscript it 4, and that then means repeat this 4 times. So that's a useful trick. And then you've got an amino group right on the end. Okay, so that's the structure of the amino acid lysine. Okay, right. The proteins that make up histone core complexes, they have lysine residues in. Okay, and some of these lysine residues in specific histone proteins can have groups added onto them, and when you add these groups on, it's going to cause the DNA to loosen how tightly it's wrapped around the histone core complex, and therefore the DNA will be more open, and that's a key word that's used all the time. And the DNA will be more open and more easy for RNA polymerase II enzymes to work its way along. Okay, more easy for it firstly to bind to the promoter region and then work its way along to produce the piece of mRNA. Okay, so you'll get an increase in the expression of the gene basically if you open up the DNA. Okay, right, so which groups can be added on to lysine residues? Well, one of the groups is an acetyl group. Okay, so let's start by discussing the acetylation of lysine residues within histone proteins. Okay, so basically you can add on acetic acid molecules, okay? So an acetyl group comes from an acetic acid molecule. So an acetic acid molecule is the old name for a molecule that would now be called ethanoic acid. Okay, so this is acetic acid here, or ethanoic acid. Okay, and this is just a 2-carbon carboxylic acid, which is fully saturated. Okay, it's the acid that is within vinegar, basically. Okay, now, what can happen is you can bind acetic acid onto the amino group of lysine residues by an amide link. Okay, so what's going to happen is we're going to take the hydrogen off the amine group, we're going to take the alcohol group off the carboxylic acid group, and we're going to bind the nitrogen of the amine group to the carbon of the carboxylic acid group. And this sort of a link here is known as an amide link. Okay, so we have then acetylated the lysine residue, because what does an acetyl group actually mean? Well, you will notice that we have not actually attached on the entire acetic acid molecule. Instead, we have attached on the acetic acid molecule, subtract the alcohol group. Okay, we've attached on this portion that I have outlined in green here. Now, this is not a molecule, okay, because this carbon needs to be attached to something else, so it's not complete unless it's attached to something else. Such structures are known as groups, so this is called an acetyl group. Okay, so you can attach acetyl groups onto amino groups within the lysine residues, and that process is known as acetylation. Okay, so we can acetylate lysine residues within histone proteins. Okay, so if you add acetyl groups onto lysine residues within histone proteins, it causes the DNA to loosen how tightly it is wrapped around the histone core complex. Okay, the DNA will become more open, and then the RNA polymerase II enzyme will have an easier job firstly binding to the promoter region of the gene within this region, and then working its way along to produce the piece of mRNA. So basically, you increase the expression of that gene that's wrapped around this histone core complex by acetylating the histone core complex. Okay, right, so that's one of the groups that you can add on to lysine residues. We'll come back to another group later on. Okay, so let's remember where we have got to with regards to the histone deacetylase. So here is our DNA, okay? Let's say that this portion here is our gene. Okay, so this is our gene here. Okay, in red. Then upstream of the gene, we have got a promoter region. Okay, so here, this is our promoter region. Okay, and we'll colour in the promoter region in green. Okay, now within the promoter region, we have got one of those specific Wnt response elements. Okay, so let's say this little region here, this is a Wnt response element. Okay, so we'll colour this in blue here. Okay, so this here, this is a Wnt response element, and remember that is a C followed by another C followed by a T, where have I got it? Followed by two more T's here, followed by a G, and then followed by WW. Okay, so you've got that little sequence here, and then what's happened is that you've had your TCF/LEF family transcription factor bind on top of the Wnt response element. Okay, then in the absence of beta-catenin, you've had one of those Groucho/TLE family proteins bind on top of the TCF/LEF family transcription factor. Okay, so this is a Groucho/TLE family protein. This that is bound directly to the Wnt response element, this is a TCF/LEF family transcription factor, and then on top of the Groucho/TLE family protein, we've had a histone deacetylase, an HDAC bind. Okay, so now what's going to happen is the histone deacetylase enzyme here is now going to remove any acetyl groups that you have bound to lysine residues on the histone proteins which this gene is wrapped around. Okay? That then is going to cause the DNA to wrap more tightly around the histone core complex, and hence this gene and this promoter region are going to end up by wrapping more tightly around the histone core complex, meaning that RNA polymerase II is going to have a harder time binding to the promoter region. Okay, and therefore you're going to get less transcription of this downstream gene. So basically, in the absence of beta-catenin, these TCF/LEF family transcription factors are assembling this complex upon promoter regions which have Wnt response elements within them, and this complex is causing a reduction in the transcription of the downstream gene. Okay, right. So now let's turn our attention to what's going to happen in the on state. Okay, so remember in the on state, beta-catenin becomes present. Okay, right. So now let's draw this picture once again. Okay, so we'll draw it here. Okay, so again we've got our gene, and I'll just show this a little smaller this time. So here's our gene in red. Okay, then we've got our promoter region upstream of that gene. Okay, and remember our promoter region has got one of these special Wnt response elements within it. Okay, so let's put this here. So here is that special sequence of 8 organic bases, which is the Wnt response element. So let me just move this up a little bit. So in blue here, this is the Wnt response element, which for short is abbreviated to the WRE. Okay, then we have our promoter region here in green. Okay, now we've still got our TCF/LEF family transcription factor bound, but now we're in the on state, and there is beta-catenin present within the nucleus. Okay, so now instead of the Groucho/TLE family protein binding on top of the TCF/LEF family protein, we're going to get beta-catenin binding there. Okay, so this is beta-catenin. Okay, now it's finally time to discuss what beta-catenin is. Okay, so beta-catenin had this fancy name that it was a transcriptional coactivator. Okay, so we now want to discuss what actually is a transcriptional coactivator. Well, basically, it is not a transcription factor. It is instead a molecule which is capable of binding to transcription factors, and in this case it's binding to a TCF/LEF family transcription factor, and it's going to change the function of that transcription factor. So previously, the transcription factor of the TCF/LEF family was decreasing the expressions of the downstream genes, okay, downstream of promoter regions with the recognition site for that transcription factor in. Okay, once this transcriptional coactivator binds here, okay, it's going to change the function of TCF/LEF transcription factors so that now they are going to increase the expression of the downstream genes. Okay, so transcriptional coactivators bind to transcription factors and change their function. So previously these transcription factors were repressive, now they're going to become enhancers. Okay, right, and the reason for this is that beta-catenin is now going to bind to an enzyme. Okay, so we'll colour this enzyme in orange here. And this enzyme is a histone methyltransferase. Okay, so this is what's known as a histone, and it's methyltransferase. And for short, histone methyltransferases are often abbreviated to HMTs for short. So this is a histone methyltransferase. Okay, right. Now, again, there is not just one histone methyltransferase, it's a whole family of enzymes, okay, but they all do the same thing, which is that they methylate lysine residues within histones. Okay, so let me just discuss the methylation of lysine residues. So let's draw a lysine residue here. Okay, so once again, here is our core amino acid structure, the amino group, the alpha carbon, the carboxylic acid group. And then the R group of a lysine residue consists of 4 methylene groups. Okay, and again I'll just draw 1 and then subscript it 4. And then we've got our amino group here. Okay, now I'm going to draw the amino group a little bit strange, but you'll see why I've drawn it like this in a moment. So remember, the nitrogen in a normal amino group has a lone pair of electrons here. Okay, right, so this is our lone pair of electrons. Now, what histone methyltransferases are going to do is they're going to bind methyl groups onto the amino groups of lysine residues. Okay, so we're going to bring in a methyl group here, okay, which will consist of a carbon atom with 3 hydrogens. And then this carbon will have a positive charge. It will have lost an electron. Okay, so remember, carbon usually has 4 outer shell electrons. Okay, but in this case it's only got 3 of its own. The other 3 come from hydrogen atoms, so it has actually lost 1 here, okay, giving it an overall positive charge here. So what's now going to happen is basically it's going to form a bond with this nitrogen, but the nitrogen is going to put in both electrons, so both of these electrons come from the nitrogen. Okay, like so. That then gives the carbon 8 outer shell electrons. However, it means that the nitrogen has effectively lost an electron, because remember, the normal understanding of a covalent bond is that 1 electron comes from each of the 2 members within the bond. So, it's as though 1 of these electrons belongs to the nitrogen and 1 now belongs to the carbon, basically. So that means that the carbon has gained an electron, which takes it back to neutral, but now the nitrogen has lost an electron, so the positive charge that was originally on the carbon has been deferred onto the nitrogen here. Okay, so that's how you methylate a lysine residue. So the histone methyl transferases are going to methylate lysine residues within histones, okay, and that's going to result in the DNA becoming more open, okay, so the DNA will wrap less tightly around the histones now, and then the RNA polymerase II enzyme will now be able to bind to the promoter region more easily, and it will be able to work its way along the gene more easily. Okay, so you'll get more mRNA being produced for these downstream genes. So overall then, in the off state of the Wnt beta-catenin pathway, you have very low expression of all of these genes which are downstream of a promoter region with a Wnt response element within it. Okay, then when beta-catenin suddenly appears, when the Wnt beta-catenin pathway is turned on, then you're going to suddenly increase the expression of all of these genes by opening the DNA at those regions. Okay, so this is how the activation of the Wnt beta-catenin pathway changes gene expression. Okay, all of these genes with Wnt response elements are going to be expressed in cells which are being stimulated with Wnt. But they're not going to be expressed in cells that are not being stimulated with Wnt. So it is the activation of these genes with Wnt response elements in their promoter regions which maintains these intestinal stem cells as stem cells, basically. This is how Wnt maintains those intestinal stem cells as stem cells, and why when the daughter cell of that results from the division of a stem cell gets pushed out of the area where it is exposed to Wnt, it's going to suddenly lose the Wnt beta-catenin pathway, and all of these genes are going to turn off, and that's what triggers it to start the process of differentiating into a terminally differentiated cell. Okay, so to summarize then, let me get my picture of the intestine back. Okay, so to summarize, you have these intestinal stem cells. These intestinal stem cells are in the right place. They live in this stem cell niche. They are receiving Wnt ligands from these PANF cells right at the base here. Okay, and these Wnt signals maintain them as stem cells by turning on the expression of these genes which have a Wnt response element within their promoter region. Okay, when a stem cell divides, it produces 2 new stem cells. One of them most likely will get to stay in the stem cell niche, whilst the other will get pushed upwards. It will then no longer be exposed to Wnt signals, and therefore it will turn all of those genes off now. Okay, so it then no longer remains as a stem cell, so it will now begin the process of differentiating into a terminally differentiated cell. It will then undergo a rapid proliferation in this portion of the crypt of Lieberkühn to produce loads of these cells that are on their way to terminally differentiated cells. Okay, then when they emerge onto the surface here, they will have completed their differentiation. They will now be in either an enterocyte or a goblet cell. These are the goblet cells, or an enteroendocrine cell. Okay, these cells will all gradually migrate up the sides of the villi to the top of the villus, where they will commit apoptosis and pass into the lumen. Okay, so that then is the continual renewal of the epithelium of the small intestine. Okay, and remember, these cells take around 3 to 5 days in mammals to get from here to the tip here.